to use this technology in different respects. So it's trying to find new biological phenomena or analyze them. We are mostly diagnostics and therapeutics. That's why I'm in the first session today. But we're also studying the principles of nucleic acid evolution, which is still, after 35 years or 30 years, something like a black box. But NGS and data that the areas we're working in is we're trying to use aptamers as inhibitors. So that's one of the first things people have done, right? The aptamers for proteins and use them as inhibitors. We're doing this. I'm not talking about this today. We have done something adaptive binding to monitor metabolism in the way that we are using this super cool progenic RNAs. We have developed light-depending systems. That's what we have done in the past 10 years. And I spoke about this a couple of times when I was here. We're doing cell surface zone analysis. So we're doing aptamers for cells. And we tackle this a little bit today. But we have done also a lot of chemical modified aptamers over the past 15 years. And that's what I'm going into today. What is it? Also, yeah.
You're the kid.
Right. That better? Let's see. Okay. So that's what we're talking about today. So put a little bit of the framework. So modifications of aptamers has been a major task in the past and 30 years too. So it started with X and A's with fluoro modification with the PUPAM and amino modifications, omit oxy and so on and so forth. I'm not talking about this today, but there have been developments in the past 10 years, I would say, that extend the base pair possibilities, like for the Yao base pairs shown here on the left with the hydrophobic base pairs, or also quick or hydrogen bonding base pairs from Stephen Banner's lab, and also, of course, the famous monomers from Larry Gold's lab, which attach something on the nucleobases itself, additional chemical entities. And we're doing something similar, which is very close to this. We're using click chemistry to introduce modifications at the C5 position of uridine, which is quite handy because the click chemistry is easy to do, it's quantitative, and it allows us to introduce many different chemical modifications. But of course, we're not alone with this. So there was a paper even before we published this by the Isaac Krauss group. They used the same methodology also with libraries, but in a very different way. So they introduced large glycan residues and try to find out where to position those that are binding to HIV-related proteins better than with just having one glycan residue. And there's a follow-up publications from Soul's group. They're using not this tiny short linker that we have been using, they're using a larger linker and attaching also glycan structures. There's Julian's and Marcel Hollenstein's group who are using the same chemistry, but they're doing a little bit things different. They're using different types of structures, like this cuban here. That's why they call this cubamers, which is very nice to sound. But they're doing the click chemistry on a nucleotide triphosphate level and then incorporating this. Whereas we're doing incorporating the EDU and then they're clicking on these things. But basically, that's the same process, right? So they're all clicked in kind of modified nucleic acids. So we did a lot in the regard of doing this for proteins. We have done it for, of course, it's a very easy model target like GFP. That was our first one. We have started in also model target, but then for the CXC09 and so on and so forth. We funded a company based on that. So they're exploiting these technologies right a bit further to get this into a diagnostic market. Let's see how that goes. We don't know. But we also did it for small molecules like THC. So this was for us a game-changing technology in this regard because we tried THC targeting with DA, 2'-fluoroRNA, and so on, and never succeeded. Then we succeeded with this entity that we introduced there, and when we found out that a couple of years ago, another group published a DNA aptamer that binds to THC, which we were never able to get. So I don't know what's the magic there. And what we're talking about today is then we moved on. So we have small molecules and proteins. So now we have a history of using aptamers for cells. So we try to address that we can use this technology for targeting cells. And when we can get aptamers, we call them clickmers, that can be also handy in vivo. And the long-term goal in this is that we the question is, can we target different receptors on the surfaces of those cells with discrete molecules instead of DNA or canonical DNA, and whether they have advantages or disadvantages when we're using them in vivo? Something we don't know yet. I will go there a little bit in our last slide. So we did the research. So the entire cell project with the quickness was started by Lucha in my lab, I think almost 10 years ago as well. And then the in vivo studies were done by David, he's here also today, and by Mayer, and they did the in vivo targeting funding we got from the ERC and the German Research Council. We had a strong support from our Genetic Resource Center at our institute. Without this, we couldn't do any mice experiments at all. Okay, so what did Muchab do? He started to generate this complicated cell scheme, and I walk you through very quickly. So it's basically the routine scheme that we're always using. We have incubation, separation, amplification, single-strand displacement. What we're doing differently here is the following. So we have a library which has the alkyne residues. Then we split them into 5 entities. We click 5 different chemicals to this, combine the library again, and then we incubate the target cells. And we do then some counter selection with alien cap cells. So the target cell is PC3, so cancer, prostate cancer. And we do pre-selection for alien caps. It's also prostate cancer, but a much more less severe cancer type. And then we did recovery of the bound cells, emulsion PCR, single-strand displacement. And then what Muchab did is, on the level of unmodified single-strand DNA, he went again to the PC3 cells, but then he took the old supernatant and went on in the cycle. That he did to prevent that we are enriching for DNA that are also binding without modifications. And because we really wanted the aptamers or the thickness to be binding depending on this. So we use these 5 different entities, so imidazole, amines, just the aliphatic, isopropyl, isobutyl residues indole, and a cyclic peptide. So that's what we clicked in. And with this, what we usually do is we ran a couple of cycles just with this enrichment profile. And so we did this 9 times, and then we do the sequencing, besides binding data, of course, but we do sequencing to look into this, and that's how our typical starting libraries look like. We have an equal distribution of G, C, and A, and then a little bit less of the T, so that's the modified nucleotide. That's we didn't do this. Reduce a little bit the amount of modifications that we have per strand because they're also costly. And we have found out that we don't need 20 of them. Sometimes a few of them will be sufficient. So we did this for 9 cycles. You see that there's a change in nucleotide distribution over the random region, which is an indication that something's happening to the library, but it's not very strongly enriched at that point, right? What we then do is we try to deconvolute a little bit. So it means we're running 3 deconvolution cycles, and that's what that means. It's illustrated here. So we're running the selection, we split it, but we then couple to one of the modifications, but we don't combine anymore. So we run 3 in this case shown, but we do it 5 different modifications. We run it 5 times individually for 3 cycles. And then we do NGS together. What it looks like this is then shown here. So that's the nucleotide distribution of then the 3 additional. So that's the starting material, and then after 3 cycles with just imidazole, distribution looks like this. Isobutyl looks like this, and so on and so forth. And what you can see is that if you do it with imidazole, isobutyl, and with ethylamine, the distribution looks fairly similar. Right? So you can see this in this area and this area. That's really mimicking kind of. And if you do it with the peptide and with indole, it's also kind of mimicking. Yeah? So it looks similar. And you can see this also if you look at the 2 families that you're getting. So you can see here that we can name this family S1 and family S2. And then if you look at the entire distribution, you can have the frequency of the family. So this is going through the 9 cycles, so it's a split combined pathway. Then we're doing the different enrichments with the different individual applications to see that with imidazole, ethylamine and isobutyl, it's going up, it's going down with the other 2. And for the other sequence, it's the contrary, so it's going up with the peptide. It's going up a little bit with the indole and not so strongly with the other 2. So what we then do is usually we take representatives of each family, so we call them S1 and S2, in the binding data. So what you see here is something that we have never seen before. We have done this split combined selects a couple of times, but if you then take one sequence S1 from the first family, so that's the binding of the enriched library just as a control. That's you're using a scrambled sequence. That's just the EDU state. You don't see binding at all. But it binds if you do imidazole, peptides, isobutyl, isolenamide. It's not so well binding because it can go. Right? But that's something, the same sequence with different chemical entities, something that we didn't expect at all. And also for the other S2 sequences, similarly, you see binding with these entities, and not so much with this one, and not so much with this one, but it was much more enriched with here. That was the winning modifications of the other one. So what we then did is we skipped this, we focused on this sequence for further investigation, and what I'm showing you is only data with these two modifications here in this sequence, because otherwise it would be just messy. So we usually then are doing is we try to find out which positions of the enriched sequence require to have modification. So the original aptamer that we selected has 8 positions in which we have the modifications in it. So we then do to find out whether they're necessary or not, and we do walking, right? So this means we exchange each position by a T, which cannot be modified, and just one T here all the time. Then we walk through and we will bind data. So that's the control of the fully modified sequence. And so we can see that this position 5, 6, 5, 6, and 37, you can have them as a T and you have still full binding. If you do anything else, you see that there is an impact in binding to a different extent. That's for the imidazole modified version. There's a similar pattern you get for the RGB modified version. And if you do just the DNA, it's not binding at all. So this means we can reduce it probably to having 1, 2, 3, 4, 5 modifications, and then we're testing sequences that have an assembled less amount of modifications. That means we have replacing more than one X via the T and test binding. So this is 5 modifications, 4, 3, and 2 modifications. So if you do this with the imidazole modified version, you see that the 5 and 4 modifications, so this and this sequence, they still bind very well. If you reduce it further, you lose binding entirely. But for the RGB modified version, the situation seems to be a bit different. So as soon as you reduce it from the full length to only having 5 or 4, there's a strong decline in binding. It's not really in line with what we have found before, but it is what it is. So that's what we have found, and what we're then doing is we're using these molecules and test for the specificity. Yeah? So we have this elaborate selection schemes where we use the alien capsid as counter-select cells. We're testing the sequences that it binds specifically to PC3 versus the alien cap, and that's something what we see. So this is the imidazole, scrambled versus the aptamer, scrambled versus the aptamer with the RND. See there's an increase in binding, and there's not so strong binding for the alien cap cells, which is fine. So we separated those binding data from the other binding data that you will see in a minute, because for the alien cap, we have to have a specific kind of media, which is different to the other cells. So we're using this so that we're doing this data extra, not to mingle it with the conditions. If you then move out to other cancer cells or the Hex cells, you see that the imidazole modified S1 doesn't bind at all to the other modifications, to the other cells. But the RGB, same sequence, but with RGB, binds also to MCF cell, which we have no idea why, but it's what it is. And if you move on to confocal microscopy data, you can see this as well. So these are PC3 cells. That's the imidazole version. That's the RGB version. You see that the red dots, that's the aptamer, that's the nucleus. The white things is a cell membrane staining. These are the controls. They're pretty clean. See both aptamers are taken up by the PC3 cells very well. And if you go for MCF7, you only see the cyclics, the peptide modified versions that are also going up into the cell, taken up by the cells. But the imidazole is not taken up. So that seems to, so seeing is believing. It's much better to have flow cytometry, but this data, but that's then a little bit confirmation that we have something that is really different, but it's just the modification that goes to the it's accounting for the difference, but we still don't know why, and we don't know yet. So we then do always, and you notice we're truncating. We're trying to shorten the aptamer as much as possible. So pigments are expensive. We'd like to order them, so we go for truncation, and what we usually do is we try to enfold and all the stuff, but it's usually not very helpful. Yeah, so what Mucho did, he cut it off the primer binding side. So this ends up with a 40 tumour, with having the 4 modifications. Then he cut it further down, but what he has seen is that the 40 tumour binds well. That's the sequence. If you truncate it further, you lose binding. And the same thing happens with the RHD, which is kind of astonishing because it has a strong, more intense binding if it's truncated. That's controlled. And if you remember, if you do it on the full length level with just the 4 modifications, we saw a severe drop in binding. So that's the full length data, and that's the truncated data, which is not expected, but that's what's happening sometimes because it can have an impact on folding, because the long sequence is longer and so on. Maybe it's stabilizing here a certain substructure that is responsible for binding. We don't know. Because that's where we are at that point. So we don't know the reason for this. We have to do structural data, but we haven't done this so far. What we then did, and that's my last data slide, David did this, so we moved on and tried to find out whether they not only bind on a cell culture, which is a nice thing to have, but if you would like to make value to it, you need to see that they're also targeting the tumor tissue in vivo at least. What we're doing in this regard is we're using nude mice models, xenografts implanted in the neck area with PC3 cells. We let them grow to a certain size, and then we inject, very pragmatic, this sequence, which is the 40 tumour with its 4 modifications and with low fortified. There's no capping, there's no peg, nothing. We just inject this in the tailway, and we let circulate for until 24 hours, and we take images. And what we see there is, so we're using this as our aptamer that's shown here. So that's the tumor area on the neck. And that's the control. That's a scrambled sequence of this, having the same amount of modifications, but the nucleotides in a different order. And then you calculate the amount of aptamer or control sequence you find in the tumor tissues versus a reference tissue, which is somewhere there where you usually don't have enough a lot of staining. What you can see at least is that we increase in staining of the tumor with the treatment. We see not an increase with the control, and you see that you're losing the staining when you go up to 24 hours. So it's going down again, which is okay. But we don't know whether that's good or bad, but we see it's staining of the tumor tissue, which gives us now enough data to move on with this. So, and this brings me to my last slide. So what I showed you is we can do the split combined click cells also on cells, which is not a big surprise, but we have seen this. We see that the thickness are click depending. They have a very strange interaction profiles. We have one sequence that binds with different chemical entities. Is at first something we were kind of, okay, what the hell is going on? We don't know. Can we use this? But, well, we also have found out that they're binding to the tumor cells in a different way. So we lose specificity with the peptide, we gain specificity with all other modifications. And we see that they're targeting in vivo. And now that's a big advantage. Because what we can do now, and that's what we will do in the next, is we will see whether we can do the tumor targeting with all different click-ins that we're using in the same sequence, which is an advantage because we can really investigate the impact of the click-in moiety. We can see whether the targeting is also tumor type specific. So reflecting the same binding behavior between the vitro and the vivo. We can investigate the pharmacokinetics more deeply because we can really pin it down to the modification we're using. And we haven't used any other modifications yet. So there's still a lot. We can use something that's very similar to imidazole, different linkers and so on. And then, of course, something we need to do and also want to do is we have selected for cells, but we have no idea what's the actual target on the cell surface. So we need to do target identification. we started this, but for the stigmas, it's difficult because they have a high background interaction. We do pull-down experiments, so we have to figure out what might be the best way to do this, but I think it's a matter of optimization. Okay, so with this, I'm done, and I'm happy to take any questions. Thank you.